The genome of the coronavirus, they are RNA viruses, positive sense, single-stranded and RNA virus. The size is about 27,000 to 30,000 of the genome. And it's clear that how it looks like. It has the spikes are there, just like the other viruses, and we will study its genome in detail. They are enveloped and spherical viruses, about 120 nanometer in the diameter. The RNA genome is associated, most importantly, with the N protein to form the nucleocapsid. Helical for the genus coronavirus and the tubular for the genus torovirus. Of course, it is associated with two different genuses, torovirus and the coronavirus. We will study them in detail also, the diseases caused by the coronavirus and the torovirus. And it is the linear single-stranded ribonucleic acid are present inside their genome. The size 27 to 32 kb, and the largest of all RNA virus genomes are present. This is again one of the largest viruses, or the coronaviruses. They are capped. Of course, they are polyadenylated with the cap, and the linear RNA at the 5 end of the genome is also present at the end of the each subgenomic RNAs. Now, the most important part of its replication, that how this kind of the special so big RNA is replicated. The replication is most importantly cytoplasmic. It happens in the cytoplasmic. There are different steps involved in the replication. First of all, of course, the attachment and the entry. Attachment of the viral proteins with the receptors. The protein of the virus which is attached with the receptors is the S protein. Later, then the virus will be entered inside the body, and the entrance is caused by the endocytosis. Endocytosis of the virus into the host cell and the fusion of the viral membrane with the endosomal membrane. Then the viral membrane will be fused with the endosomal membrane, and the genome will be, of course, released inside the cytoplasm. Synthesis and proteolytic cleavage of the replicase of the polyprotein will be done. A double-stranded DNA genome is synthesized. We have already studied that the virus is single-stranded RNA plus sense genomes. However, in the replication, the double-stranded genome is synthesized, while double-stranded RNA is transcribed by providing the viral mRNA genome, and the synthesis of structural proteins will be encoded by the subgenomic mRNAs. After the replication, the package is material, and it has to be done by the assembled in the capsid. Packaging, budding, and assembling at the membranes of the endoplasmic reticulum and the intermediate compartments to the Golgi complex, because all the processes are happening around inside the cytoplasm of the cell. Then the release of the new virons by the exocytosis. Here in the diagram, it makes it clear the whole replication process in the cytoplasm. First of all, the attachment and the entry inside the host. The receptors are there. And the S protein, this is the S proteins. S proteins is going to attach with the receptors, the spike proteins, and then it will go inside the body. After going inside the body, it will be uncoating there, and genomic RNA will be released in the cytoplasm. The genomic RNA will be just like the mRNA, the translation of open reading frame, and the open reading frame are the two kinds of the open reading frame, 1A and 1B. Then the RNA-dependent RNA polymerases will act on its transcription, replication to make the double-stranded RNAs. And similarly, the rough endoplasmic reticulum of the host, and then there's Golgi apparatus, and then the vesicles will be formed, and then the virus will be packed inside the capsid, and it is the complete mature virus which has to be exit from the body, and it will be done by the exocytosis. The entry is through the endocytosis as far as the coronaviruses are concerned, and of course it will be exit from the cell in the exocytosis process.